Today I'll be discussing my thesis entitled Examination of Potential Elicitors of Operant and Respondent Behaviors in Smokers. Thank you all for attending, and a special thank you to my committee members, Dr. Carolyn Cohn and Dr. Susan Schneider. In the United States, over 45 million adults smoke cigarettes. There's an extensive range of adverse health effects related to cigarette smoking, some of which include heart disease, stroke, and certain cancers. Given this information, it is easy to see why 23 million adult smokers reported attempting to quit smoking in the year 2010. The overall message is this: smoking is very bad for your health. To date, the most successful smoking cessation programs involve a combination of approaches. These include a combination of antecedent and consequent manipulations. Nicotine replacement therapies and behavioral interventions such as contingency management programs are the most effective at promoting abstinence from smoking. However, they do not have lasting effects. In fact, long-term abstinence rates range from 20 to 30% following the completion of these types of cessation programs. Low long-term abstinence rates among smokers who have previously quit smoking suggest that we have more work to do. Understanding both the consequences that maintain smoking behavior as well as the antecedent variables that influence tobacco use may prove valuable not only to smoking cessation interventions, but to our basic understanding of smoking behavior. While no one claims to have the complete answer, we can look to the drug abuse literature on operant and respondent conditioning to learn more about how operant and respondent learning processes relate to this type of behavior. Stimuli present prior to the occurrence of behavior and subsequent consequence can, over time, exert control over the frequency of that behavior. These stimuli come to function as discriminative stimuli, or SDs and S-deltas. SDs in a person's environment increase the probability that a response will be emitted in its presence. This is due to a history of reliably being present prior to the delivery of a reinforcing consequence. For example, if a person is in the presence of an unlit cigarette, this may serve as a cue that lighting the cigarette will be followed by a reinforcing consequence. Over time, in the presence of that unlit cigarette, a person might be more likely to light the cigarette. Bradley et al. Observed increased attentional bias when smokers observed smoking-related stimuli as opposed to neutral stimuli. Also in this study, smokers who had previous multiple attempts to quit smoking had greater attentional bias for smoking-related visual stimuli compared to non-smokers and smokers who had never attempted to quit smoking. It's possible that the increased attentional vigilance is due to a history of smoking-related stimuli being present prior to cigarette consumption, and as such, the smoking-related stimuli, or SDs, are exerting a type of stimulus control. Basic research performed by Siegel et al. May help explain the process by which stimuli in the environment can elicit physiological responses in the body. In 1982, Siegel and colleagues tested heroin overdose in rats who had never received heroin before and heroin-experienced rats. When each group was placed in a novel environment, they found that the dose of heroin was lethal for the inexperienced rats. For the heroin-experienced rats, it depended on where they received the drug. For these heroin-experienced rats, the dose was lethal in a novel environment, but was not lethal in an environment containing conditional stimuli related to heroin ingestion. Previous research suggests that drug administrations provide multiple opportunities to pair environmental stimuli, such as people, objects, or activities, with a drug's pharmacological effect on the body. In 2005, Siegel discussed potential compensatory behaviors related to drug consumption. He suggested that when people repeatedly take a drug in a particular environment, certain aspects of that environment may become conditional stimuli for conditional compensatory physiological responses to that drug. For example, if a drug tends to increase heart rate upon administration, then in the presence of conditional stimuli in the environment, the body might reduce its heart rate in preparation for the impending drug. Siegel suggested this conditional physiological compensatory response may account for drug tolerance and withdrawal effects. In 1991, Rose and Levin discussed the role of respondent learning as it relates to smoking. Previous research suggests that nicotine is the key addictive component in cigarette smoke. Rose and Levin found nicotine self-administration by cigarettes presents a rich context of sensory cues that accompany the act of ingestion. Nicotine is a key sensory cue in cigarettes because of its distinctive taste, aroma, and irritant properties. These sensory cues include the irritant scratch in the throat that's provided by the cigarette smoke, or the taste of the cigarette smoke. So it's possible that the taste and olfactory cues become secondary or conditioned stimuli due to their previous association with the effects of nicotine. Cue reactivity research exposes individuals to drug-related cues using in vivo sessions. This can include watching somebody smoke, vividly imagining situations related to past drug use, audio, visual, or pictorial stimuli, while tracking participants' self-reported, physiological, and/or behavioral responses. Traditionally, cue reactivity research is focused on conditioned cues most proximal to drug administration, such as a lit cigarette or pictorial stimuli of drug paraphernalia. Conklin in 2006 suggests that distal cues, such as environmental stimuli that are not closely associated with smoking, are also key components in cue reactivity. Bradley et al. In 2003 used a visual probe test while measuring reaction time to determine that smokers tend to fixate on smoking-related visual stimuli for longer periods of time than non-smokers do. Conklin et al. In 2008 found that participants self-reported cravings when looking at both proximal and distal stimuli. They found they were also the first ones to suggest that drops in CO might function as precursors to the administration of smoking cigarettes. Machado, in his unpublished thesis, experimentally assessed whether or not smokers' CO levels changed as a function of the stimuli that they were looking at. In his study, Machado used the images from Bradley et al. 2003 and found that when smokers viewed smoking-related visual slideshows, that smokers' CO levels dropped. This systematic drop was not found in non-smoker or control participants. Since the primary measure in this study was carbon monoxide readings, I'd like to take a moment to discuss this measure further. It turns out that carbon monoxide readings are often used as a primary dependent variable in smoking cessation research. This is partially due to convenience, as CO readings are not very invasive, and also because CO readings provide a direct measurement of CO in the lungs. A subset of researchers have identified several variables that affect CO readings, which is important to address if CO is going to be a primary dependent variable. False positive CO readings, or readings that incorrectly identify levels indicative of recent smoking, can be produced when a person has excessive levels of hydrogen in their body. This may incorrectly be identified as CO due to the monitor's sensitivity to hydrogen. While excessive hydrogen levels are typically limited to people who are lactose intolerant, another more relevant source of influence on CO readings may be exhalation duration. In their study, Rafe et al. Systematically manipulated the duration of exhalations in their smoker participants. Readings in the fast exhalation condition, in which participants were instructed to exhale into the monitor as quickly as they could, produced readings that were 30% lower than those obtained when participants were instructed to exhale slowly. This has major implications for smoking-related research in general. But specifically when abstinence from smoking is evidenced by low CO readings, then a smoker may manipulate these readings by simply shortening their exhalation duration. Of even more relevance to my thesis, it is possible that when specific patterns of CO readings are found and the length of exhalation duration is not held constant, that the compensatory-like responses obtained are actually due to changes in exhalation duration. So this brings me to the purpose of my thesis study 1 and study 2. I replicated and extended the Machado study and added a number of integrity measures to strengthen the methods, as well as control for a key threat to the validity of the study. Specifically, the study controlled for total exhalation duration, which allowed me to parse out whether or not there was additional evidence for the presence of physiological compensatory responses in smokers that view smoking-related stimuli. Also in Study 2, which will be discussed a little bit later, we examined whether or not the systematic patterns observed in Machado were due to gross changes in exhalation duration by removing the total exhalation duration control. So 6 people participated in Study 1, 3 smokers and 3 non-smokers. Participants that produced CO readings at or above 11 parts per million were classified as smokers, and participants that produced CO readings of 3 parts per million or below were classified as non-smokers. Smokers that at baseline produced CO readings between 4 and 10 were excluded from the study. All participants were randomly assigned to one of 2 groups in which they experienced baseline 2 non-smoking conditions denoted as B and 2 smoking conditions denoted as C. The order of the sessions was counterbalanced for the second group. 2 experimenters were present for each session. The primary data collector, or experimenter 1, was blind to the order of experimental sessions. The secondary experimenters' duties included filming CO readings, performing IOA agreement checks, and ensuring procedural integrity throughout all sessions. Visual stimuli in the study was adapted from Bradley et al. And Machado. As an example, here is a sample of some of the images used in the Bradley et al. Studies. Smokers were able to identify the ones that they felt were smoking related or non-smoking related. It's images like the ones depicted here that are used in all smoking and non-smoking slideshows, which I will discuss a little bit more in a minute. Our primary dependent variable was carbon monoxide, which we measured using the Covita Pico Plus Smokalyzer Breath CO monitor. We also obtained blood pressure and heart rate measures. Lastly, we administered the FTND and had participants fill out a demographics form. Pictures were obtained for all heart rate and blood pressure readings. Experimenters were also trained to use the specialized audio software program Audacity, which we used to precisely measure exhalation duration. Experimenters were allowed to conduct sessions when they had reached a sufficient speed and accuracy using the Audacity program. IOA was calculated for 33% of CO readings, heart rate, blood pressure readings, and CO duration exhalation. So I wanted to take a minute and discuss a few features of the Audacity program that I mentioned in the previous slide. This is a screenshot of the Audacity program. The Audacity software was used to convert audible CO reading exhalations into voice waveforms, depicted here in blue, which allowed for the visual analysis and precise measurement of exhalation duration. I want to draw your attention to some of the capabilities of this program. This is the waveform produced by an actual CO reading, and as you can see, we can clearly see where the first and second beep occur, followed by an extended beep, which indicates that the device is ready to begin the reading. Audacity also allows us to manipulate the y-axis to increase the size of the waveform, which allows us to more accurately visually detect the start and stopping points for duration. Also, we can measure the amount of time the participant is exhaling by highlighting the waveform and counting the seconds depicted in the top red box. This general procedure was used for Study 1. After completing the informed consent, the primary data collector modeled how to use the CO monitor. Participants were then asked to take 3 CO readings approximately 30 seconds apart. These 3 CO readings were then averaged to determine the exhalation duration cap. Heart rate and blood pressure readings were taken, followed by a picture sorting task and a control slideshow, a 5-minute control slideshow. During the control slideshow, primary data collectors had 5 minutes to determine the exhalation duration average that was used in all subsequent readings. Going back to the CO readings, as an example, if someone exhaled for 5 seconds, 8, and another 5 seconds, their duration cap would be 6 seconds for all subsequent readings. In all subsequent CO readings, when they reach 6 seconds, they would be asked to quickly remove the device from their mouth and discontinue exhaling. The session would conclude with a post-sorting test, as well as carbon monoxide, heart rate, blood pressure readings, and the FTND. For all smoking and non-smoking conditions, participants were welcomed. We obtained physiological measures. They viewed an individualized slideshow consisting of the images selected in baseline as either smoking or non-smoking related, and then we concluded with another set of physiological measures as well as the FTND and demographics form. Results from Study 1. Across all participants, longer durations of exhalations resulted in higher CO readings, and this is consistent with the Rafe et al. Literature. Among non-smokers, there were no distinct patterns of responding in any condition, that is, no systematic patterns of pre to post increases or decreases in any session, and no appreciable differences in responding from the control participant. Additionally, for the smokers, we did not find any systematic patterns of responding. So let's look at the individual data on one participant for some further explanation. This is the actual data from participant S003, which was a smoker. For the top left graph, the y-axis represents the CO reading and the right y-axis represents exhalation duration. Along the top we have pre or post, and along the bottom we have session labels. For each of the 2 smoking conditions, for each of the 3 smoking conditions, we don't see systematic changes or pre to post slideshow drops in CO readings. For both graphs, a gray background denotes smoking conditions. So as you can see, the smoking the smoking exhalation duration remained constant throughout experimental sessions, and looking at the bottom graph, there were no systematic pre to post decreases in any of the experimental sessions. Because differential responding as indicated by drops in CO among smokers in the smoking conditions did not occur in the first 3 smoker participants, we decided that the key baseline exhalation duration manipulation would be removed for study 2, and another 5 participants would be entered into the study. The purpose of study 2 was to see if the results obtained from Machado 2011 were related to gross changes in total exhalation duration. For study 2, we used the methodology with all integrity measures in place, except exhalation duration and baseline, and across all experimental sessions was no longer held constant. Instead, participants were instructed to exhale slowly and completely into the device until all of their air has been expelled. As mentioned before, we had 5 participants in this study, all of which were smokers. For this study, we recruited from the mental health population. This population was chosen because there was a higher prevalence of smokers in the mental health population compared to adults not diagnosed with mental illness. Doing this helped alleviate some recruitment difficulties. As with study 1, across almost all participants, longer durations of exhalation resulted in higher CO readings, with the exception of 1 reading for 1 participant. As with study 1, there were no distinct patterns of responding that occurred for any of the smoker participants. As you can see with this graph, the participants' natural patterns of exhalations varied quite a bit during sessions. As a reminder, the gray background indicates smoking sessions. If you look at the second graph below, you'll see 1 instance in which the participant had 2 pre to post drops in CO readings during a smoking slideshow. There were no other consecutive pre to post drops in CO readings for any other participants in any other smoking slideshow. A second experimenter took pictures of all CO, heart rate, and blood pressure readings to allow for inter-observer agreement to be calculated. IOA was calculated for 33% of sessions by taking the number of agreements divided by the total number of agreements plus disagreements multiplied by 100 to obtain a percentage. IOA for exhalation durations was defined as experimenter 2 identifying a CO reading that was no more than plus or minus 1 second from the CO reading duration identified by experimenter 1. IOA between experimenters 1 and 2 was 84% for exhalation duration, and disagreements never exceeded 1.5 seconds. IOA for CO readings and heart rate and blood pressure readings was 100%, meaning that the photos of heart rate and blood pressure readings matched those written on the datasheet by the experimenter 100% of the time. The first 6 participants in this study, 3 non-smoker and 3 smoker participants, showed no discernible patterns of responding in any of the experimental conditions and no discernible differences from data obtained from the control participant. That is, the results of Study 1 did not replicate Machado 2011 findings and provided no additional evidence for the presence of conditional physiological compensatory responses when total exhalation duration is controlled. At this point, the data could have been interpreted in one of 2 ways. The characteristic patterns observed in Machado 2011 were a function of changes in exhalation duration, thus controlling for exhalation duration eliminated the possibility of producing drops in CO, or the characteristic patterns observed in Machado were not replicable and may not exist. For that reason, Study 2 was designed to examine whether patterns observed in 2011 could be recovered when exhalation duration caps were removed, which would provide evidence for the role of operant learning. 5 smokers completed Study 2, and only 1 participant in 1 smoking condition produced 2 consecutive pre-to-post decreases in CO. Similar to Study 1, each participant provided variable pre-to-post changes, but with no clearly recognized pattern of responding. This is in contrast to the results obtained by Machado 2011, in which each smoker participant produced pre-to-post decreases in CO readings for each smoking condition. Although using 2 different populations in a study is not inherently problematic, it may have been so for this study for 2 specific reasons. First, researchers have known for many years that a subset of patients diagnosed with schizophrenia do not orient toward relevant stimuli and fail to provide and failed to properly habituate to relevant stimuli over time. If this were the case for most or all of the participants in the current study, this could have caused significant problems given that the independent variable was smoking or non-smoking visual stimuli, to which participants were expected to orient and attend. Moreover, recently, researchers have identified ways in which psychiatric drugs may disrupt learning processes. Dion et al. Found that SSRI and SNRIs disrupt discrimination training tasks in rats. Other studies have found the same patterns relating to other psychotropic drugs. These findings have not yet been extended to the human population, but the possibility that psychotropic drugs can influence learning processes introduces the possibility that learning may have differed between Study 1 participants and the participants in Machado 2011. Although results from the current study do not provide evidence supporting Machado 2011, further research is warranted. For example, in light of the effects that psychiatric drugs and serious mental illness mental health diagnosis may have affected attention and learning, future studies should enroll only typically developing adult smokers. If possible, in addition to CO monitor readings, researchers may want to obtain blood metabolite readings that may provide a more sensitive measure of CO readings. That's it. Thank you.